Okay. So first, so today what I want to do, I want to go back to the exercise a little bit, show you some of the answers. I looked at the stuff you sent me, so not everybody has sent results yet. I think you should at least try to send it, but I will not grade this exercise as I explain. So don't worry if it's wrong or not good. Yeah, I can start by showing you. Okay, so no. And after, yeah, so just so you know what we are doing today, so I'll do this. I will show a few things, some background geological information for next week, for next Monday's discussion. And then you will have a lecture from René about the Great American Biotic Interchange. Okay. So first, I want to show you some of the results that I got from you. So some of you even managed to make a movie, and I can show it to you. So this is from Pia. Pia, you don't mind if I show this?
No problem.
Okay. So I'm gonna share my screen. Okay, so this is one of the results from Pia. Very nice presentation. And so here you see, let me remove that. Do it again. So next time, Pia, you can put some music, also it will be more dramatic. So here, yeah, you see this thing coming in. And then 40 MA, 35. Somehow it starts to become straight. And then here at 28, everything is closing in. And then here you get a contact at 12. And interestingly, in this reconstruction, you get contact here with this block, but also with the block below at the same time, more or less. Yeah, and that's what you wrote nicely here. Yeah, so this is nice. This is nice. I think there were still some issues. No, it's okay. I think this is almost, yeah, it's perfect. So you have at 38. But no, maybe the only thing that is not correct here is that between 38 and 28, this is the blocks. The two blocks here are rotating with this block here. So I think that should not be the case. And then, but then it's fine because at least you have this straight arc at 30, and then it turns between 28 and 30. So this is okay. Now I will show you another one that we got. Yeah, so here from Nora. Nora, can I show it?
Yes, sure.
Okay, let's see what we got. So this is, so here things are moving here with this block. And then it's more or less the same as the other one with the same results. Okay. So it looks like, yeah, you worked with the same files. Now let's see. Okay, well, that's okay. I will just show you now how things should have been done. So you can see my screen now. Okay. So if we, so the first question was to put everything together like this. And I think most of you reached that point without any problem. Then it was okay, using this initial rotation file that you get from G-plates, what is the collision age, the connection, and so this was about 3 million years, okay? And then the next part where things getting a bit more difficult is when we started to rotate the blocks. And so here, so what we discussed is to make a reference to this one, 2013, I think, this block here, because it's not rotating so much during the entire period, and it makes a good reference. And so we had this first block, 2014, and this is the one that is that rotated actually counterclockwise, if I remember correctly. And so we want to make it rotate. So I think most of you got this right, but I'm just showing how I do that. So here I go to the reconstruction sequence, and then I look for my block 2014 with respect to 2013. This is the one, and here I can edit that sequence, and here I will just add one. So we go back to 25 million years, right? No, sorry, no, that's not the way to do it. Sorry, I think I'm gonna confuse you here, but okay, it's good to have the sequence on the side. No, here we just work with the block. And so we go here, 25. Okay, and then we're gonna rotate that block. So I choose the tool for rotation here. And then I need to look for the pole. So enable pole here. And I bring this pole here on my area. And then I put the pole where I want this thing to rotate. And then I take this tool here and I can rotate it. And so here I will rotate it clockwise, 25 degrees, okay? So that after, when it goes from 25 MA, no, how much was it? 20 degrees? 25 degrees, yeah. Okay. Okay, and then I apply this rotation, okay? And so now at 25, the block rotated 25 degrees.
Can I ask something?
Just a second, let me finish. And then when I continue, when I go back to 0, it rotates then counterclockwise 25 degrees. Exactly what I wanted. Okay, yes, go on.
Why did you set the pole of rotation in between the two plates and not exactly in the center of the plate that we want to rotate?
So in fact, yeah, good question. So in fact, we have a choice here. We can put it more or less anywhere. We know the block rotated 25 degrees with respect to north, but we don't know where the pole of rotation is actually. And so to know where it is, it's more because I want to get a straight arc at the end. So that's why you can rely on paleomagnetic poles, but you also need to have other information to reconstruct your arc. Okay, and you see here I put it on the side, so in the end it's relatively straight, and it looks good to have a straight arc. Okay.
Okay, thank you.
So the other thing you can notice is that the other block, so this one, 2015, has moved also with this block because it's connected to it, okay? But if you look in the, actually that's not what we want. We didn't want this block to rotate 25 degrees during this time.
It should rotate 10.
It should rotate 10 clockwise, but with respect to north, not with respect to this block, okay? So to do this, this is where you can go. So I selected this block. You can go on this file actually and set the reference for this block to 2013. So this is 2015. Dun, dun, dun. I think it's this sequence. here. And here on the top, I can set it to 2013. Apply. Okay, and now it's back here. And so you see, only this block, 2014, is rotating, and the other one has not moved at all. Okay, we will deal with it later. Okay, well, or maybe I will just do it now. I don't have to do the full reconstruction. I think that will be enough. So now let's deal with 2013 for the rotation. So I go to 25 MA. Okay, and here this block should have rotated. Oops. Yeah, this block should rotate 10 degrees clockwise. Okay, so pole, where is my pole? Here. So now I'm going to put the pole here. It doesn't really matter actually. You will see why. And now I want to rotate 10 degrees. Oops, so what's going on? Maybe I have the wrong. Oh yeah, I'm here. I'm moving plate 2015. Oh, that should be the case. Alors, attends, I'm having a small issue here. So this is a pole. I select the right plate.
Okay, I know which probably it's because you just made a change to 2015. This is the same I had the other time.
Oh yeah. Well, maybe I need to change the other file. Yeah, because he's using an old pole. Okay.
You need to go into the rotation file somehow.
Yeah. No, it's because here, I think it's because here I have I still have 20. No, this is 2014. I still have a reference to 2014 on these guys here. And that should be 2013. I'm not sure actually, but I think that's the problem.
Yeah. I think the same was with me.
Yeah.
Yeah, I also had a problem where it wouldn't move.
Well, I hope this is it.
Let's hope.
So now
Time now is 107.
Oh yeah, okay. It's me as I struggle through this thing. So the pole is okay, and now, yes, that was the problem. Okay, so now I'm rotating clockwise 10 degrees. No, counterclockwise 10 degrees, right? Because I wanted to rotate clockwise after.
Yeah.
10, okay, apply. Okay, and all right. And now you see it's really not well positioned compared to the other block. So what I'm going to do is move the block. So I'm at 25. And here to move it, so you go here, you unclick enable block, and then you can move that block the way you want. So it's already rotated, but I just wanted to align with the other one. Okay, apply. Okay, and that's it. Okay, so now we have this block.
Beautiful.
It's nice. Okay, so now the last thing I wanted to show, and then we can move on because we don't want to spend the whole time on that, is how to connect. So this last block here, 2031, to the rest, because it has nothing to do. This is actually the block that is connecting with South America. So where the Gabi is going on. And so it had nothing to do attached to North America like this. So this is completely wrong. And so to do this, I go in the files again. And so 2031 is here. and I will try to connect it, so all of them now, this time, I will, to 2013, actually. Easier than that. Okay, apply. And here, I do the same. Check. Apply. And here, the same. Okay. And so now it should, yeah, okay. So now it's connected. Actually, it's connected to this block here. Okay, so it's but it looks very bad. And so I will position it at 25 like the other ones. Okay. And so what is the rotation of this one between 25 and 0?
20 clockwise.
20 clockwise. Okay, so first deal with the rotation. So select the block, enable pole, put the pole, let's say here, and I will rotate it 25 clockwise. So 20 clockwise, right? 20. Okay. Apply. Did it work? It didn't work.
It's the same then. It must be something.
Ah, yeah. Something wrong here.
It must be again in this file.
Ah, yeah.
2031.
No, it should be fine.
It is 2031, no?
Yeah.
The one we selected. Huh. Strange. Exactly the same shit.
You had the same?
Yeah,
okay.
This is when we went into the rotation file. So
okay, let me try again. Somehow there is a bug here. 20 degrees. Huh? Okay. 20. Maybe something is wrong.
2031.
Looks good.
Looks all good.
Yeah.
But look at the whole sequence. There's
Then
nothing
it's
showing.
the same as your case, right?
Yeah.
It's not changing.
You see, there's in the post. There's nothing
Yeah.
in here.
Yeah, yeah.
So there's
Should
something
be.
not.
Yeah, it's not connecting to the.
And do you need to apply this, the reconstruction sequence? Do you need to?
Well, it should be okay.
If you close it, what happens?
Let me see. So this is okay. Yeah, close it. To see.
And maybe you should say save here. You see the rotation file. Let's say save.
But it works without saving it. So I don't think. Well, we can try. I did it before and it worked without saving it. So let's try it. No,
Let's try.
Maybe just move on, I think.
Yeah.
We run out of time.
Yeah, yeah. Okay, sorry, I cannot fix this bug right now.
I'm sure this on Monday.
But okay, then the other thing, so here it's not rotating. Maybe there is a... Okay. Yeah, the other thing then, the last thing you would do, maybe it will make everything work, is move that thing down there so that it aligns with this. Let's see if I can do it, but yeah, apparently it doesn't work. Also, it shouldn't be at 32, but at 25. Okay, apply. No, yeah, I don't. It should find the pole sequence. Sequence crossover is not yet automatically synchronized. Yeah, I don't know what's going on. Anyway, you see here, so this is at 25, so it should be here. Now we have this straight arc. And so this is 25. So 28, there was still another rotation, a big one actually. And so if you really straighten the arc here, you can have the collision occurring here at 29. Yeah, I don't have time to move it, but so depending on the connection, on the reconstruction that you do and where you put your blocks, you can have the collision so very early or much later if these blocks are just skimming across it. So the message in the end was that you don't have, I mean, the precision of this reconstruction is not really high, especially if you don't have marine magnetic anomalies around it. And so that other constraints are very important to have, okay, from geology and especially sedimentary records. So let's try to share that. So now I want to talk about next Monday's debate. So yeah, so all the papers have been taken, and now I want to talk about this paper here from Montes, which is a lot of geology, and this is to help the presenter of this. So one thing that is used in this paper is to date the connection. No, not really. To date the connection between the two blocks coming in by looking at the sediments. So the sediments that are collected in basins between these blocks, they actually tell you where the source rocks are coming from. So in this case here, you see the two blocks are coming together. And when they are very close, then the sediments coming, the red sediments from North America will mix with the blue sediments. As soon as you see these red sediments in your Panama Basin sequence, you are sure that North America is not very far, okay? And it's the same if you have a basin in North America and you start to see blue sediments coming in, then you are sure that Panama is not very far. Okay, is it clear? So this is a classic technique, and it's called provenance technique. So then the thing is that the color of the sediment might not be a very good argument, not very diagnostic of a continent. And so what is diagnostic, more diagnostic, is can be the mineral composition of the source rocks. It can be also some kind of fossils that you find, that have been transported. And one thing that is now being used extensively is to look at the age of the source rocks, okay? And so this is what is done in the paper that will be presented. So what people look at is actually the age of zircons. So zircons are minerals that have uranium and lead, so lead produced by uranium in them. And so zircons, when they get old, they have a lot of lead and they get damaged. And so you can actually tell them apart from their color. So young zircons and old zircons. And so, but you can also date them. And so we'll go back to this in the class later, but there are techniques to date that precisely. And so basically what you do is you date the grains of sands. And so this is just, I don't want to go too much over this paper because it will be presented next Monday, but this is exactly what's being done here, is that so you're looking at basins here in North America, okay, and looking at the composition of the grains. And you see here that there are some young blue arc rocks that are at some point being transported over. And so if you look at a sedimentary sequence, so from older to younger, at some point, you see the appearance of these blue younger zircons. And so that means that Panama is not very far. And so you are close to the collision, okay? So it's very simple. Technically, what happens is, so here you have the age of the grain. So this is one sample. So they dated 108 grains, okay? And this is the age of these grains. And sometimes you have 1, 2, or up to 15 grains within an age bin. Okay, so this one has a lot of ages, around 100 million years here, or 80 million years, that are more like from North America. But it also has these young ages that you do not find in North America, and you can only find in Panama.
South America.
The blue grains come from Panama.
But the black is South America.
Yeah. Did I say Panama?
North America.
Oh, North America, yeah, sorry. Okay, any questions on this before we move on?
Yeah, from my side, why do only the young ones from Panama and not the old ones?
Yeah, so no, you can get old zircons also from Panama. That's correct. And then you cannot tell them apart from the ones that are also old and coming from South America. So if there was not these young rocks in Panama, you couldn't tell the difference. And that can be a problem sometimes. I mean, you need to have a diagnostic rock that is providing some grains. Otherwise, you cannot see it. Good question.
So basically the rocks which formed on Panama are younger in general than those on.
Well, there are some younger rocks in Panama that you do not find on North America. Yeah, and so they are diagnostic of Panama. Yeah, but there are also older rocks on Panama. That's a good point. Yeah. Okay.
Okay, thanks.
So we can discuss this more on Monday, maybe.
Well, I hope we have time and it will be presented. But I just wanted to give some geological background so that it's clear.
Yeah, let's talk a little bit about South America today. And in the end of this presentation, we'll come to the Great American Biotic Interchange. So this topic we'll discuss on Monday. Yeah. So a few words about South America. You see here in a satellite image, it is the 4th largest continent on our planet, on nearly 18 million square kilometers in size. And we have 3 major topographic features. We consider South America on a large continental scale. These are obviously the Andes, which run along the western side of the continent all the way from the north to the southern tip, yeah, and reach an elevation of nearly 7,000 meters at their highest point. And then we have highlands here in the Guinean Shield, yeah, Venezuela and northern Brazil. And we have these Brazilian highlands here, yeah. And in between, we have these lowlands, including the Amazon basin. So this is the largest area of South America, these lowlands, and the Amazon River is the largest river of the world. Yeah, so we have very relevant topographic features. And here in the bottom of this figure, you see, of this slide, you see a cross section where this is visualized for this area south of the Amazon basin. So we have the Paraguay River here as one of the big rivers. Yeah. So, and you see that if you look at the satellite image, we have very different coloration here. We even have snow here, so white areas. We have these dry areas or these brown areas, which are arid, and then we have these dark green areas, which is rainforest. So how can we explain this distribution? And we need to look into climate. Yeah, if you want to learn something about the distribution of ecosystems and the distribution of biodiversity, then we have to do something about climate. Does everyone know of you what is Hadley circulation in the intertropical convergence zone is? So this is basic climatology, and I can skip this if you know these things.
I do not know about this, so I would like to hear about it.
Okay, so I mean, this is just general climatology. So think about the Earth facing the sun, and between 20 degrees south and north, the sun hits the Earth's surface at the right angle, and therefore this is called the thermal equator. This part of the Earth is heated much more substantially than the areas north and south of this. And this results in an energy surplus in this portion of the Earth, which needs to be transferred to the north and the south. Yeah. And because of this transfer of heat, we drive circulation cells. What causes winds? Wind is an air movement from high to low pressure, and high pressure is caused by heating. Yeah. So at the equator, we get the convergence of two of these cells. Yeah. So we heat the surface, and go back. And then at the equator, we have rising air. The air cools, condenses, can't hold moisture, and rains out. And then at about 12 kilometers, we have the troposphere ends and the stratosphere starts. There, the air column can't rise any further. It's deflected to the north and south. Yeah. And then because it becomes drier and heavier, it descends. That's where we have the subtropics and mostly desert environments. And return. So this is one of these are the Hadley cells. They are a critical component, and where they converge, this is what we call the intertropical convergence zone. This is a very wet area. Yeah. Another part of the climate system of South America is the South American summer monsoon. The monsoon is a circulation system which acts between ocean and land. Yeah. And when we have the Southern Hemisphere summer, this is December, January, February. Yeah, the sun hits the Earth's surface south of the equator. Yeah, and this causes heating and the formation of clouds, which we call deep convection. Yeah. And these winds, when they cross the equator, coming from here, the Earth is rotating to the east. And then when they cross the equator, they get deflected because we have a circular Earth. And this is the summer monsoon. So you get wet climate here, and you see that this portion of South America stays dry, and this portion stays dry during this time. And then we have winds, westerly winds, coming from the south, and they build another convergence zone here. So this area is also dry. You know? So if we then combine these two concepts, the intertropical convergence zone and the monsoon, we get our seasonal climate. So think about these Hadley cells converging at the point where the sun reaches the Earth pretty much at a right angle. What happens to the location of the ITZZ over the course of the year? Any idea? Okay, obviously not. So we will see this. Because we have seasons, because the Earth is on an angle, you know, and the sun is circling. Over the year, the Earth is circling around the sun. Different portion of this Earth's surface will receive solar radiation at a different angle. And so this 90 degrees incoming solar radiation will be in the north of the equator, in the northern summer, and south of the equator, the southern summer. So it shifts its position and it crosses the equator twice. Yeah. So this is what we see here. This is the distribution of rainfall over the year. There you see in the northern summer we have heavy rain, northern South America, and now in the southern summer we have this monsoon and rain mostly south of the equator. Yeah. So this is associated with the seasonal shift of the intertropical convergence zone and the intense South American summer monsoons in the southern hemisphere summer, which is the boreal winter. But you also see that certain parts remain pretty dry. This corner here, this area here, yeah. And you see that only a few places are ever wet. That means they receive rain most of the year. This is mostly the area around the equator. That's where you have then rainforest. So there's another phenomenon when we talk about the climate. Has anyone heard about the El Niño Southern Oscillation or El Niño phenomenon?
Yep.
Yes or no?
Yes.
So you can explain it, yeah?
Well, I've got some broken memory bits that that I could share. So I remember it has something to do with the direction or the strength of the warm currents in the Pacific, if I'm not wrong, and they have an influence on where the rain of this planet is going to pour down. And I feel like something changed accordingly, but I don't get it too good.
Okay. Anyone else with an idea? Okay, I'll try to explain it briefly. I mean, this is a major. I mean, we can have a climatology lecture on this phenomenon alone, but our class tries to cover some climatology because this is so essential to understand. You know, not topography alone and tectonics is influencing the biodiversity, is also climate. And the El Niño Southern Oscillation is a phenomenon which affects the entire tropics, not just South America, also Southeast Asia and also Africa. Yeah, and it's associated with the Walker circulation. So we talked about the Hadley circulation, which goes in the north and north-south direction, which we call a meridional circulation. Here we have a zonal circulation, so they go along a zone. Yeah, that means from east to west. And what drives this are gradients in temperature and pressure. So normally, you know, because of the rotation of the Earth, you have winds going from east to west. Yeah, we call these winds trade winds. Yeah. And what they do, because they drag along the surface of the Pacific Ocean, they pull the water somewhat towards Australia, Indonesia. So in fact, the sea level is slightly higher here. And because this is an area which then is receiving a lot of solar radiation, it warms and we get warm water here and a lot of convection. So this is why Southeast Asia is very wet. We call this region the Indo-Pacific warm pool. It's the warmest area on the planet, and there's always normally a lot of rain. Yeah. Because of this dragging of the ocean surface, the ocean also has this internal circulation. So on the surface, the ocean is pushing towards, towards the west, but then it's downwelling off the coast of Australia. Sorry, it's hard to do this without a pointer. So here, yeah. So here the ocean goes down, then it flows on the bottom, and it's doing what we call upwelling here. And you get cold water up here off the coast of Peru. Because you have cold water, there's little little evaporation, yeah, and we have dry conditions. That is the reason why we have the Atacama Desert, for example, yeah. So you have basically a mirror of the atmospheric circulation in the ocean, yeah, a reflection of that. This is the Walker circulation. Now you can have two modes. One where this circulation is intensified and you get very wet conditions. This is what we call La Nina. And then you have really droughts in Peru. And then once in a while, between every 2 and 9 years, this system becomes unstable. You know, so the water is basically like in a bathtub, it's flowing backwards. Yeah. So because we have a slight difference in sea level, this is not stable anymore. And then what you get is a shift of warm water to the central and eastern Pacific. And it rains more here then. This is when we have these catastrophic fires in Indonesia, partly in Australia. So this is when we have dry conditions on this side of the Pacific. And we have very wet conditions in Peru and Galapagos and elsewhere when we have heavy rains. And at the sediment record, we see these changes. We can reconstruct El Niño patterns by looking, for instance, at lake sediments. And we see that there's, when we have an El Niño in Peru, we have a lot of erosion into lakes. And if we have normal conditions, there's much less of this material. Yeah? So we can do this. But this is such an essential mode of climate variability. So it's a natural phenomenon, which basically results in dry or wet conditions on a decadal timescale or subdecadal timescale. So taking all these things together, we can look at the average distribution. Was there a question?
Yeah, a question. Okay. I just wanted to know where these instabilities come from. Are these only nature given, or is there any explanation? Thanks.
Oh, this is a very good question, and it's probably, I can't give you a conclusive answer. There's a huge amount of work into modeling this phenomenon. Yeah. It is something which must be related to how strong the circulation is. And the circulation, again, is influenced by other issues related to the distribution of heat. And it becomes really complex when we consider the global circulation pattern. So you need a climate model to do this. You know, there's not one single mechanistic explanation here. It's just that you must imagine if the trade winds are not strong enough, they can't maintain this piling up of water in the West Pacific, and it just moves backwards. Yeah. And with that, once you create whatever is causing a weakening of the trades, and you can have a positive feedback, it just starts and you can't really stop this phenomenon. And it also takes a while to really set this in again. So El Niño lasts mostly then a year or so, yeah. So it's it takes a while to really get this driving again. But it's not very clear what's really causing this the thing to shift. Okay, so if we look then on the general climatology of South America, which is enormously important to understand the patterns of vegetation types and the patterns of biodiversity, we see on the left side the temperature annual average temperature distribution. We see we have large red areas, which are the deep tropics. There we have average temperatures of around 27 degrees or so. Yeah. And then we see some steep gradients in temperature associated with the highlands of the Andes, where we get actually, if you go up to the Andes to 4,000 meters, very unpleasant. It's like just above freezing point. Yeah. But in the south, you also maintain relatively cool temperatures. This is Patagonia. Yeah. If you look at the distribution of rainfall, we have highest rainfall here in the Amazon basin, and we have a high rainfall here and there. Yeah. So we will see how this is reflected in the distribution of vegetation types. We also have dry areas here, which I pointed out earlier. What is relevant about South America is that it's, on the one hand, has the wettest place on Earth, which is Colombia, with almost 12 meters of rain a year. And then it's the Atacama Desert, where at certain years it rains not at all. Here you see a climate diagram for this very wet area in Colombia, where every month it rains over 400 millimeters. So this is almost annual precipitation in Brandenburg. So it's extremely wet. And that's why we have a rainforest here on the western side of the Andes. So we can translate all this climatology into a climate map. You don't need to look into this now in detail, just look at different coloration. It reflects essentially the combination of temperature and rainfall. And not just mean annual rainfall. What we do, if you look into the ecological relevance of climate, we have to look into the distribution of rain over the year. This is the rainfall seasonality. Yeah. Is there a dry season or not? And if there's a dry season, how long does it last? These are very important issues which influence the vegetation. Yeah. So an ever wet rainforest does not persist if there's a dry season longer than 3 months. Yeah, and a dry season in the tropics is a month with less than 100 millimeters of rain. So we can translate now this map here. Yeah, this is a map of climates. And we look here at the map of biomes. Biomes are large-scale vegetation types. So they have nothing to do with the species composition. They just reflect the type of vegetation. Is it a forest, a rainforest? Is it a savanna? Is it a steppe, a desert? Yeah, you will find these biomes on every continent. I mean, not on every, but the biomes are a global phenomenon. They are not, have nothing to do with the species composition. Yeah. They're more like reflecting what we call the physiognomy, the morphology of plants, etc., the adaptation to the climate. And you see that we have forest here, rainforest, yeah, and here. And we have these dry areas here and there. And again, forests are in the wettest part. So here we see the distribution of biomes on climate space. So we plot precipitation against temperature. And rainforests can only persist, just this is a rule of thumb. It should not be below 20 degrees Celsius in a month of the year. And it should be about, let's say, 2,200, 2,500 millimeters of rain per year. Yeah. If it's colder, you get a different forest type. If it's drier, you get some forest which is deciduous. That means it sheds the leaves during part of the year. Yeah. So if you're drier and colder, we get different systems. So here a number of local names for these ecosystems, which you should at least have heard about. Yeah, if you talk about South America. If we want to understand South America, we need to know, or we need to acknowledge that there are different types of ecosystems. So tell me what you think, if I ask you, what do you think about, what's the first thing pops up when somebody asks you South America? What would you think? What is the first thing which comes to your mind? Just say something. Rainforest, rainforest. 3 more answers. Just what do you think when you think about South America? First thing which comes to mind. I would say Amazonia. Yeah.
Amazon.
Yeah, perfect. That's what most people associate with South America. And what you need to realize is that there's a great diversity of ecosystems. Of course, Amazonia is very important. and is large, for sure. It's the largest rainforest pocket on the planet. But we have a situation, for instance, that in eastern Brazil, we have an area which is called Caatinga, which is pretty much not nearly a desert, but in a very arid area. And we have these gradients where we go from Amazonian rainforest into this Caatinga and also the Mata Atlantica, the Atlantic rainforest, which is here along the Atlantic coast, is directly bordering this Caatinga. And here in the middle, between the Mata Atlantica rainforest, Amazon, we have the Cerrado. This is a very large savanna ecosystem. Yeah. So we have ecosystems which you may associate with, let's say, Africa, where you think about savanna. But this savanna is very large in South America indeed. We have other ecosystems which you think about, like the steppes of North America or Asia. This is the Pampas here. This is a wet grassland, and then the Patagonian steppe is a dry grassland. Then we have these highland ecosystems. Along the equator, we call them paramo. They're somewhat wetter. And on the Altiplano plateau, we call it puna. It's dry alpine grasslands. And then we have also these savanna ecosystems in Venezuela, where we call them llanos. And here in northern Argentina, Paraguay, and Bolivia, there's the Chaco, another savanna ecosystem. So this reflects the different distribution of rainfall and temperature. Yeah. So you can only understand the biodiversity of South America by appreciating that there are different ecosystems. Yeah. So if we look now at the map of biodiversity, we see that the most diverse areas are, in fact, as we would expect, the rainforests, yeah, the Amazon basin, but also the Atlantic rainforest here. Yeah. This is for tetrapods, so all animals with 4 limbs. Mammals, amphibians, reptiles, and birds. Okay, some more data on biodiversity. If we compare the tropics in terms of species richness, number of species, America or South, or what is called Latin America, or let's say the neotropics, it's the biogeographic region, stands out. It is not rivaled by Southeast Asia or by Africa in terms of species number. This is from a very recent paper by Peter Raven, one of the probably the greatest living botanists, and they have compiled a new list of species, and they say that nearly 120,000 species of vascular plants occur in the neotropics. Yeah, this area which is shown in purple. 9,000 species of butterflies, 4,600 species of birds, nearly 4,000 species of ants. You can read these numbers, and if you compare them to Africa and to Southeast Asia, we will see South America or Latin America has most in every case. Yeah. So it is the most biodiverse place on Earth. So climate is one observation. We have a diversity of ecosystems. Yeah. We can look in more detail, and I recommend you, which I did in the first lecture on biodiversity, look at this website, biodiversitymapping.org. You can download these maps and you can play around with them and you can measure this. I'm not showing here numbers for birds. I'm just showing you dark red is the highest diversity and blue is the lowest. What you see is 2 things or 3 things maybe here. You see one thing is the Amazon basin is rich, and then the foreland of the Andes. Yeah. This is here the Madre de Dios River. This is here the richest bird area on the planet. Yeah, so there's Manu National Park. This is, there you have an enormous diversity. You have also here in Ecuador, this very rich area in the forelands of the Andes. Then what is also relevant is the highlands, the Venezuelan highlands are here, and the Mata Atlantica, the Atlantic rainforest. Yeah. You look at mammals, we see a similar pattern here, this area in Peru, southern Peru, and here along the Maranon, one of the tributaries of the headwaters of the Amazon. This is the richest area. And again, these highlands, the Guinean highlands, Venezuelan highlands. Yeah. If you look at amphibians, same, the Western Amazon basin, Peru, Ecuador, but also the Atlantic rainforest. Yeah. If you look at plants, we see a similar pattern. Yeah. In fact, there are 3 so-called centers of vascular plant diversity, so areas where we have over 5,000 species of plants. Yeah. And we have the Costa Rica Choco area, which is Costa Rica and Panama, and northern Colombia. Then we have the Andes Amazon, and we have in Brazil, the Atlantic rainforest. So there's a concentration of a lot of biodiversity. And you also remember we talked about this concept of biodiversity hotspots. Hotspots are not simply the places where we have the highest biodiversity. Hotspots include a large number of endemic species, so they only be found in this region. And because the biodiversity hotspot concept is a conservation concept, it implies that most of the habitat of these areas has been lost. So the diversity is a high level of threat towards extinction. And you see that the Amazon basin is not a designated biodiversity hotspot because we still have not lost more than 70% of the Amazon. However, the Cerrado and the Atlantic forest are biodiversity hotspots because the Atlantic forest is reduced to over 90% by now. So even though it holds an enormous amount of endemicity and high species diversity, it's very unlikely that those species will persist because they're only remaining in fragments. The Cerrado is largely converted to rangelands and agricultural fields. So what you had as a natural savanna in South America is also largely gone. And you see essentially, if you go from Mexico or actually from Texas or Colorado all the way to Patagonia, the entire West Coast and almost all of Mesoamerica and the entire Antilles have been largely anthropogenically overprinted. So all this area still holds a lot of biodiversity, but it is largely anthropogenically degraded, and the ecosystems have been destroyed and converted to some kind of fields and other things. Yeah. So this is important to keep in mind. Okay, shifting from biodiversity to biogeography. So the distribution of species which are confined to certain areas or concentrated to certain areas. South America is part of the what we call the neotropical botanical realm, which is shown here. It's number 3. And you see that it extends all the way to the southern tip of Florida and a little bit into Mexico. Yeah? And that the southern area here of Patagonia is not part of it. It's part of Antarctic, the Antarctic realm. This is because they are very different climatic conditions. And you remember these Gondwanan floral elements. We have these Notophagus, for instance, the southern beaches, which we also find, for instance, in New Zealand and so on. Yeah. If you look at the animals, it looks slightly different. The southern tip of Patagonia belongs to the neotropical region, according to Wallace. Yeah. And if you look into more detail, we see that Wallace divided the neotropical region into 4 subregions, which he called the Chilean region, which is essentially the cold and dry areas. So it includes most of the Andes, Patagonia, and these dry areas in Argentina and Bolivia and so on. Yeah. The neotropical region, here we have the largest area is what he called the Brazilian region. This is essentially the Amazon basin, the Atlantic rainforest, and large parts of the Cerrado. So they are one area. And then he distinguishes what he called the Mexican region, which is Mesoamerica, all the way to the Isthmus of Panama, from Mexico here, and the Antilles. Yeah. So they all have their specific fauna, yeah, even though they share largely many families and so on. Let's take a closer look into the transition to the Nearctic, the region to the north. Yeah. We talked about these transition zones when I talked about biogeography, and you remember Wallace's line. this was defined as a sharp boundary between the Oriental and Australian region. We have no equivalent of a line here, yeah, between what we call the Nearctic region and the Neotropics. We have a transition zone, the so-called Panamanian transition zone. But we can narrow down the point where we can say, here is the start of the Neotropics, and here is where we say the temperate boreal elements phase out, which is actually in Mexico. This is the Isthmus of Tehuantepec. I don't know if you've heard about, but this is the closest area between the Gulf of Mexico and the Pacific. It's wider than the Isthmus of Panama. It's also not a plate boundary, but it is the narrowest point. And what is relevant is here that in this area, this is where the Cordillera, where the mountain range is interrupted. So all this land here is below 225 meters in elevation. And you see here, you have a lot of mountain range. So many species find their range limits here, and many species only extend towards here. We see this also reflected in a climate. If you look at temperature, you see here that these highlands are cooler than the lowlands here. And then the Yucatan Peninsula and so on, it gets really warm. And it's even more clear if you look at the distribution of rainfall. There's sharp rainfall gradients. Yeah. So you go from an arid region here in Mexico to a very wet region. Yeah. And so the Isthmus of Panama, which is the collision zone of South and North America, is not the point where we say this is where the neotropical biogeographic area starts. It starts farther north. Yeah. Should keep this in mind. Yeah. And we actually see, if you look at the satellite image, these dry conditions are reflected in this kind of brown tone, which reflects grasslands and deserts actually, yeah. And then we go into forest. And people had looked into the transition or this for different groups of animals. Here I show a map for these small mammals, rodents, moles, and hares, and here for bats. And they basically plotted the distribution of multiple species from the tropics and from what we call the Nearctic, and looked where are the range boundaries. And you see a transition zone here, which is relatively narrow, and then you see here the Neotropics starting. And the pattern between these two groups or these groups is very similar. Yeah? So you have here a transition zone where bats phase out, and here the neotropical bed starts. And you can see that it's largely this isthmus. So Yucatan Peninsula is already the neotropics. And it's fascinating to see that Wallace predicted this without the modern tools. He did not have databases, he did not have distribution maps, and he drew a line which is pretty much consistent with modern taxonomic and phylogenetic work is predicting. Yeah? So Wallace's map is still valid. So let's talk about a little bit more about the biodiversity of this region before we come to the American interchange to have an idea. Yeah, so if we look about the flora, so the neotropics now. So we look all this area, let's say, which starts in this part that we just talked about. So southern Mexico, essentially. This is the neotropics. We have 25 endemic plant families, yeah, 3,500 endemic genera. In Amazonia alone, we have 2,500 species of trees. Can anyone say how many tree species we have in Germany? 30 is a good number. I would say 30 is pretty good. Maybe if you look at a few more, maybe 50, but definitely not more. That's a good number. Thank you. So here, if you go into the Amazon, you make a plot on 1 hectare, you find more species, you find over 100 species in a hectare, more species of trees than in all of Germany, as an example. So it's an enormous species diversity. We have several endemic plant families, just a few to mention here, the Bromeliaceae. You may use them in your living room as a plant. So I think the pineapple is part of this, the Cactaceae. So everything, if you think about a cactus, if you see it growing elsewhere, it is invasive. Yeah, Cactaceae are endemic to Americas. Yeah? What we have here, we have similarly looking species, they are the euphorbias. Yeah? So there are no cactaceae in Europe or in Africa, naturally. And we have most relevant cultivated plants coming from America. So the Germans like potatoes or French fries, they come from South America. Tomatoes, maize, cacao, amaranth, sweet potatoes. So there's a huge diversity of plants which were cultivated. If you look at fauna, we have several bird families, in fact, over 30, which are only occurring in the neotropics. And some of them, which you should remember if you talk about America, are, for instance, the hummingbirds, the colibris. Then there are the tanagers and the toucans. Can anyone tell me what potential analogs are for the hummingbirds or colibris or the toucans in what we call the paleotropics, so the Africa or Southeast Asia? Any idea what would be an analog or similarly appearing species which have taxonomic no relationship with them? No. Thanks for the suggestion. So the hummingbirds are basically, we have convergent evolution in Africa are the nectar birds there. They look almost the same and are also very diverse, same size, and feed on nectar, yeah, as the colibris do. They're the nectar birds, you can look them up. And for the toucans, which are frugivores, so they eat largely fruits and dispersed seeds, so they're very relevant. We have the, what are they called? No, I forgot it, of course. In Africa and Southeast Asia, we have the hornbills. Yeah, you can look up the hornbills. You will see they have similarly large shaped bills, but they have nothing to do with toucans. Yeah. Okay, so let's look at some specific fauna for the herpetologists. Yeah, so we have amphibians. You heard about the poison dart frogs, Dendrobatidae, over 200 species endemic to South America. You may have heard about the glass frogs, so frogs which are almost translucent, so you can see the interior, their organs from belly. This family contains about 150 species, and then there are other species. 96% of the amphibian species which occur in the neotropics are endemic to this region. Yeah. They are all confined to there. And among some reptiles, which you might find cool or interesting, the caimans are endemic to the neotropics with 6 species. The Anolis lizards, one of the largest families of lizards, occurs exclusively in the neotropics and a little bit in the southern coast of South America and North America. Then you have the iguanas, here the rhinoceros iguana shown on this picture, and the boas, the subfamily Boine, yeah, with the anaconda as an example. So a lot of endemism. And now we go close to the American interchange. South America is a continent separated from Africa about 100 million years ago. And as such was isolated for, let's say, at least about 90 million years. It was essentially an island like Australia. Yeah. And it had not direct land connections to any other continents. And the result of this, there was the evolution of certain groups which are confined or which were like evolving in this landmass alone. There's a book about it by this paleontologist Simpson. It's called Splendid Isolation, and it discusses this phenomenon. Yeah. And we have 2 major groups of South American mammals. These are the Senatra, yeah, is shown here. They include 2 orders, which is the Pilosa and the Cingulata. So the Pilosa are the anteaters, and the sloths shown here, yeah, with 10 species. So not very rich in terms of species richness. And the Cingulata, the armadillos. Yeah, there were more sloths in the past which went extinct though. And then we have the South American or the American marsupials, which are sometimes called Ameridelphia. This is a paraphyletic clade, so it's just a concept to put this into some order. They include the opossums. Yeah, this is the richest group, the order Didelphimorphia with 110 species. And you see one opossum here. And the shrew opossums here, a small group of these insectivores. If we look at the origin of this group, the Sinatra, we see that they originated pretty much, if you see this split here in this phylogenetic tree without confidence intervals, about 100,000 years ago, based on a molecular clock, which fits pretty well with the separation of Africa and South America. And we see then the divergence of this group between about, let's say, 70 and 60 million years ago. Yeah. So it took a while before they diverged. We have two other groups which are relevant for South America. These are the New World monkeys, Platherini, and the cavimorph rodents. Yeah, and the cavimorph rodents are a group which are also only found in South America. They include, for instance, what I show here, the capybara. They are what you have at home, maybe guinea pigs or chinchillas, pacas. They are all these very interesting rodents. We have about 230 species of them and about 175 species of these New World monkeys. And here we also see that the fossil record and the molecular clocks are very close, actually. And they show us that the invasion or the, let's say, the arrival of these species, because they did not originate on, or they have a common ancestor from Africa, came about 45 to 70 million years ago. Something around 35 in the center. You see here these stars show fossils. Oops, it's too fast. But essentially they have a very similar timing of arrival. And the idea is either they rafted, we've talked about rafting before, yeah, floating mats of debris or vegetation, or there's an idea which is posted in one book, The Monkey's Voyage by Ellen de Quiros, where there is an idea that perhaps there was some traversing of the Atlantic possible. I used G plates to measure the distance here at 45 million years. It's still 1,500 kilometers, so it's pretty significant distance. Yeah, that of course extended further. And there's an idea that there perhaps were actually some little islands in the Atlantic, which are gone now, and that there were some stepping stones. Yeah. But in any case, here the fossils and the molecular dating point to a similar timing of invasion, which requires an over-ocean dispersal. Yeah. And with the arrival, we have a rapid diversification of these groups. In fact, these cavimorph rodents probably induced a lot of extinctions with the local opossums, yeah, because they had similar niches. So here we see the separation of Gondwana. Yeah. And you will see soon when South America and Africa separate. And you can now appreciate the long time South America was an isolated landmass. That's when we Sinatra evolved, yeah, when these South American marsupials diversified and these other arrived. Yeah. And then we have the collision. And obviously that's the main debate. Did it happen only at 3 million years ago? Did it happen earlier? Yeah. And so to understand, we've talked about topography of Southeast Asia. I mentioned a few things related to the climatic patterns. We saw a lot of gradients in terms of rainfall. We saw that we have a very large number of different ecosystems and biomes there, which can explain all the biodiversity. We've talked about the isolation and in situ speciation on the landmass. But the biodiversity of a region is not just influenced by speciation and extinction. It's also influenced, as we know, by the theory of island biogeography, by immigration. And one of the important things, if we talk about continents, is what we call biotic exchange or biotic interchange. It's essentially the exchange of faunas, floras, when continents come very close together, yeah, or collide. Yeah. And this biotic interchange is decisive in terms of assembling biodiversity. Yeah. In order to know when this happened and how it happened, we need robust reconstructions of dispersal pathways. That's what you tried with this G-plates exercise. Yeah. Here on these figures, we see different paleogeographic reconstructions since the late Eocene. Yeah. And we see something here we haven't talked about yet. We might, I think, we probably hear about this in one of the papers. It's the concept of Garlandia. So there's an idea that there was once, 30 million years ago, already a potential land bridge almost connecting South America to something where we are close to, I think, second here, I have to move something. No, I don't know how to do it. So this land bridge was almost close to what is now Florida. Yeah. But we see in this reconstruction that these authors assume that the closure of the isthmus happened only about during the Pliocene. Yeah. So we need robust reconstructions in order to understand this faunal exchange. We can look at sea level, and you see on this map, this is the red areas denote exposed land during the last sea level low stand. This is 20,000 years ago, the last glacial maximum. And you see there's nothing which builds a land bridge here. Yeah. You see a huge land bridge between Asia and North America, this is Beringia, and we had a faunal exchange here. A lot of immigration of species came, including humans. Yeah. We also see a connection of, let's say, Sumatra, Borneo with the mainland of Asia, and there was exchange. And also here, New Guinea with Australia. But nothing like here. The only thing which you might want to remember is that the island of Trinidad was connected to the coast of Venezuela. Yeah, this is the only thing. That's why Trinidad has some interesting endemics, since it's isolated for about 10,000 years. But for the American interchange, sea level at this time plays no role. So we have to go back. Yeah. On a global scale, we just talked about the Pleistocene. You've seen the sea level map. There was exchange, or there are three global periods of biotic interchange: the Pleistocene, the Pliocene, and the late Miocene. Yeah. Of course, in the Pleistocene, it was sea level. And in the Pliocene here, it's assumed that we had this great American biotic interchange. You see that we have two arrows here. One, which relates to rainforest taxa. And you see we only have an arrow into one direction. Why? Because there's no rainforest or no tropical rainforest in North America. So these guys could only go into one direction. Whereas for species which occur in highlands, and montane habitats and in dry habitats such as prairie or savanna, they could move into both directions. However, you remember when we talked about corridors, Simpson, this paleontologist, separated corridors into different types. And he proposed that even though you can have a corridor, it acts as a filter if there are certain conditions such as a climate. And we have here actually that the Panama Isthmus acted as a biogeographic filter. Even though it was a land bridge, it did not allow all species to traverse because the conditions we are not allowing this. It was too wet, the ecosystems were different, and these species didn't find the right food sources, etc. Yeah. So you see that a number of species from North America were stopped here and couldn't traverse. So they never invaded South America. A large number did it though, including rabbits, foxes, bears, raccoons, weasels, cats, horses, tapirs, peccaries, camels, deer. What you see here is that a large number of carnivores made this. And carnivores, of course, can often have a higher ecological flexibility than a herbivore, if it's particularly confined to specific foods. Yeah. On the other hand, we have only a few species from South America, which made it into North America. We have the porcupines, yeah, in German Stachelschweine, I don't know, a French word, maybe porcupine, no. I don't know. Then we have glyptodons, these are extinct species. We have the armadillos, yeah, so glyptodons are related to them. Armadillos are Gürteltiere in German. Then we have the ground sloths, they went extinct at the end of the Pleistocene, and the Virginia opossum. Yeah. Most of the primates didn't make it, and all these carnivore rodents didn't make it. Yeah, the anteaters and so on didn't make it because it's probably too dry up there. Yeah, and of competition with other species. So what we have now in South America is what you consider neotropical fauna, maybe, and think, wow, the jaguar, it's so specific, yeah. Or the llama, it's so specific. Yeah, they are very recent arrivals, and they descend from mammals which were able to cross this land bridge, this filter. You see here a list of species which occur in South America, and they all descend from ancestors which invaded this region during the Great American Interchange. So it was an asymmetric exchange. It was not proportional what happened in terms of immigration. And the reason, one of the reasons which is assumed, is not just the filter, but it's also that there was less, or that these species from North America could outcompete the species present in South America. Yeah. Any idea what the ancestors of the llama were? It were the camels. They went extinct also, so they're related to the camels, and they were camel species in North America which went extinct 10,000 years ago. Okay. The last slide, I'm just mentioning that there was a mass extinction event at the late Quaternary, which is related to the arrival of humans in both North and South America. So there was a lot of megafauna which is completely gone. Yeah, you see here Glyptodont, and here one of these ground sloths. So we don't find those anymore, but Simpsons and the paleontologists who worked on the Great American Interchange worked also with these species because they originated in South America when it was isolated as a landmass. Okay, that's for the lecture. I hope you got enough background to understand a little bit more about the biodiversity and the Great American Biotic Interchange. And unless there are any questions, we see you guys next Monday for a turn of presentations. Thank you.